Hello and welcome to this video on Mendel's dihybrid cross. This is an important topic. I will discuss this in this session with respect to its definition, methodology, findings and their interpretation, and the law which is derived using these findings. The law is called Law of Independent Assortment. I will discuss that. So this is a very important topic in classical genetics. In this slide, I will define the dihybrid cross, explain the selection of two distinct traits and their contrasting phenotypes, and describe the selection of pure breed plants for the dihybrid cross experiment. So let us define the dihybrid cross. It is a cross between two plants carrying pure contrasting phenotypes of two distinct traits, which produces a dihybrid. This is known as dihybrid cross. For this experiment, he selected pea seed color as one trait and shape as the second trait. For each trait, he selected contrasting phenotype. For color, he selected yellow and green. Yellow is dominant, green is recessive. For shape, he selected two contrasting phenotype, round versus wrinkled. Round is dominant and wrinkled is recessive. So for his P1 generation, he selected pure breed plant which for two traits. So he selected pure dominant, round and yellow color seeds, pure dominant, yellow and round for P1 generation, and the recessive was pure recessive was green and wrinkled. So these plants were considered or were used in P1 generation. I will explain the experiment in the next slide. In this slide, I will explain the method of dihybrid cross and the outcome from this cross. So once he selected the pure breed two traits seeds, yellow and round and green and wrinkle, these were P1 plants, pure two traits plants. He made a cross and he observed the outcome. In F1 generation, all plants were yellow and round, showing only one phenotype which is dominant, and the recessive phenotype, green and wrinkled, were suppressed. So even in dihybrid cross, he was able to support law of dominance. However, he raised the same question, and the question was, what is the fate of wrinkled and green recessive phenotypes or traits? So to address that question, he self-crossed these F1 dihybrid plants. So these F1 dihybrid plants were used as P2 generation plants in the next generation. For this experiment, he selected F1 dihybrid plant as P2 generation plants, and when he made the cross between these two dihybrid or self-cross the dihybrid plants, the outcome was 9 were yellow and round, 3 were yellow and wrinkled, 3 green round and 1 green wrinkled. So basically here recessive traits are reappearing, means they segregate and reappeared. So it supports law of segregation. So even in dihybrid, he was able to support law of dominance and law of segregation. Now there is one more thing to note here, that the shape is not tied with the color and vice versa, means they are independent of each other. That is why you are getting the ratio 9:3 to 3:1, means they are independent. Genes do not interact. You can also say that there is no interaction between two different genes. This supports their independent assortment, and based on these findings, he proposed the law of independent assortment. However, you should know what is the basis of 16 here. What is the interpretation? To explain that, he used the law of probability A, and the second he used the same principle that genes are in pairs, and using that law of probability he proved that only 16 combinations are possible of the offspring from a dihybrid cross. So but I will use here Punnett square method to explain these findings. So for interpretation of dihybrid finding, it is important that we know the genotype of the dihybrid or F1 plants. So we will start with the P1 generation plant, which were pure yellow and round. These were dominant. So write capital Y, capital R, genotype for dihybrid or two traits, pure plant, and for recessive, pure recessive, YYRR, all small letters. Make the genotype of the gametes. Only one type of gametes is possible from this parents, so this will be capital Y, capital R, and this will be capital small y, small r. So the only genotype of F1 plant is dihybrid, capital Y, capital R, small r. Using this genotype, I will use Punnett square method to explain the dihybrid findings. But for using Punnett square, you should know how to make gamete genotype from a given for a dihybrid plant. So in the next slide, I will explain make how to make the gamete genotype from this dihybrid genotype. In this slide, I will explain how many types of gamete genotypes are possible from a dihybrid such as capital Y, capital R, small r. So this is a dihybrid. There are two genes Y and R, and both are hybrid. So this is an example of dihybrid. Now you have to write gamete genotype. You have to, gametes are haploid, and you start from the left hand side. The first gene is hybrid, so split into two. Split Y, capital Y from small y, and come to then the next gene which is also hybrid. If it is hybrid, then split again and put capital R here, small r here, capital R here, small r here, and then you put the combination capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. So from a dihybrid genotype, four types of gametes are possible. So using these gametes genotype, we will explain the ratio 9 is to 3 is to 3 is to 1 using the Punnett square method in the next slide. So let us interpret the finding 9 is to 3 is to 3 is to 1 using Punnett square method. I have shown here that four types of gametes are possible from each parent. So four types here and four types here. So 4 times 4 is 16. So make 16 small squares. These squares represent nothing but these are the only 16 genotype possible from this dihybrid cross. So only 16 possible combinations of genotype are possible in a dihybrid cross. That is why you see 16 there. Mendel used law of probability to prove 16 type of combinations. So put the combination of these genotype in the corresponding square and interpret the finding. So wherever you have at least one capital Y, one capital R, that will be yellow and round. Where you have at least one capital Y, both small r, there are three wrinkled and yellow. And wherever you have at least one capital R and both small y, that will be green and wrinkled. So these are three, and this is pure recessive. So this supports law of segregation, and this also is 9 is to 3 is to 3 is to 1 supports law of independent assortment. Means genes for different traits do not interact, they are independent of each other. If you see this is also proved by their monogenic ratio. They maintain monogenic ratio close to 3 is to 1. For example, shape 12 yellow and 4 green, it comes out to be 3 is to 1. For the another trait, shape round versus wrinkle is also 3 is to 1. So even in dihybrid cross, these two different traits, they are maintaining their monogenic ratio, suggesting that there is no interaction between different genes. This based on these findings, he proposed law of independent assortment. So law of independent assortment was proposed based on the finding from dihybrid cross. It states that the factors controlling different traits do not interact and assort independently. This is called law of independent assortment. So in summary, Different factors or genes do not interact with each other. They follow law of segregation and law of dominance. However, they segregate but assort independently, and this is known as the law of independent assortment. Thank you very much for watching this video. I request you to like it, share it, and subscribe it. I will be posting more videos on Mendelian inheritance, so keep an eye on the channel. Until next time, you take care. Bye bye.